Good afternoon, good morning, good evening, wherever you are listening to this. Thank you for listening. Really excited about our conversation today. There's been a lot of talk recently about peptides. You guys have probably heard me post about peptides. Certainly, a very interesting topic to a lot of people, but important to realize that peptides are not the only thing that we can use to positively influence our health, and that there's quite a range of compounds that do exist to help us promote our health. And there's one particular type of compound that I think a lot of us think about, spend time using for our patients, but may kind of struggle to see real results and may struggle to kind of separate signal from noise. And that specifically is antioxidants, right? We've all kind of heard about antioxidants. We know that they're important to help our cells recover, to age better, but it's pretty complex to say which antioxidants to use and how much and for how long. We even have started to understand that overusing antioxidants can be negative. One question I've often had is how can we thoughtfully use antioxidants in a way that strikes the right balance? And in that regard, it's been a real pleasure for me to get to know the founders of Pathway Biosciences. They're based right here in Denver, Colorado. They've come up with a pretty interesting compound that can activate antioxidant pathways in a really unique and balanced way. And there's some pretty interesting clinical trial results that they're going to share with us today that kind of maybe suggest a different way in which we as clinicians can think about using antioxidants in our patients. Brooks and Alan, thank you so much for joining. Why don't you guys just briefly do introductions and then we can dive into some questions?
I'm Brooks Hybertson. I did my PhD in analytical chemistry, so I'm kind of the pretender when it comes to the biology side of it, medicine side of it. But that was a long time ago, so I've learned a lot in that time. And we founded Pathways Bio 12 years ago. And our chief science officer, Dr. Joe McCord, was co-founder. And he was the discoverer, along with his thesis advisor, of superoxide dismutase. So we're very proud to have him on board with us.
Awesome.
Sure. Yeah, I'm Alan Gao, and I have a PhD in molecular biology, and I'm one of the co-founder for Pathways Bioscience, and I'm the CEO for the company right now. At the same time, I'm also a professor at the CU Antioxidants, and I'm directing a genomics core facility and studied on the genomic side of the disease, including aging.
Thank you so much for joining us, Alan and Brooks. And thank you, by the way, in the past for, it's actually been cool for me to visit Dr. Gao's lab and see some of the amazing research he's doing there that involves PB125 as well as other pretty cool research that he's doing. Let's dive right into some of the key questions around antioxidants and PB125. And I know that PB125 acts on a pathway known as NRF2. Can you share with us how NRF2 relates to antioxidants and why NRF2 might be related to aging, and then also how your compound PB125 impacts all of this?
Sure, you want to start on that first,
Maybe, yeah. Well, do you want to get started, or I can chip in for?
I wasn't sure where to divide things up. As you were mentioning, one of the interesting ways that the body makes and processes oxygen radicals is with enzyme systems and not necessarily consumable antioxidants that we're all familiar with. One of the things we would like to think about is how do those enzyme systems get controlled and how can they be increased, and what happens in aging with some of these systems. NRF2 is a transcription factor that binds to what's called the antioxidant response element in a wide variety, a huge number of different genes to help increase the expression of those genes, and then usually leading to more enzyme activity downstream from that. And so when we were thinking about ways, this happens in the body all the time, NRF2 transcription factor, there's always active sensing, oxidative stress, because it's typically, am I there?
Yeah,
okay.
Yeah, go ahead.
So NRF2 is typically targeted for degradation by an inhibitor molecule, but that is sensitive to oxidation. So when the KEAP1, which is the molecule that's sending NRF2 for degradation, is oxidized, NRF2 can be released and bind to its antioxidant response element in the nucleus and turn on gene transcription. So it's always at work in your body, and the foods you eat, the exercise you get, it's all part of helping maintain that system. And so we're looking at ways, and other people as well, looking at ways to help influence that because it can decrease with age. And so we spent a lot of time developing PB125 and learning what kinds of foods and dietary ingredients can help influence positively and support the NRF2 pathway.
Let me add a little bit to what the Brooks just described. Because NRF2 is really one of the body's primary regulators for cell's defense. And when it's activated, it moves into cell nuclei and switch on hundreds of genes that help protect the cells. Those genes support antioxidant defense and detoxification pathways, mitochondrial function, protein quality control, all kinds of things to protect your cells, and even the resolution for inflammation. So rather acting as an antioxidant itself, and NRF2 tells the body to produce more of its own protective enzymes. And the PB125 we developed as a NRF2 activator, it's designed to stimulate the naturally protective response when your body needs it. So as for why NRF2 declines with aging, what you mentioned, and the evidence really suggests that it's probably like in three aspects of the following. First, it might be a contributor of aging. Lower NRF2 activity leaves cells less able to handle oxidative stress and inflammation and other forms of cellular damage, which can really accelerate age-related decline. And second aspect is it is also the consequence of aging, chronic stress, epigenetic changes, and age-related disruptions of cellular signaling can all reduce NRF2 activity over time. And third, it might be a marker of aging because a diminished NRF2 activity often reflects, the overall health and resilience of the cells in your body. So activating NRF2 may really help, restore cellular resilience and improve the body's natural defense system. But, we are scientists, right? No, it's probably not an anti-aging switch. Our goal is not to keep NRF2 permanently turned on, rather to restore appropriate and well-regulated activation so cells can respond effectively when they're under stress during the aging process.
Got it. Super helpful. One of the things that is true about PB125 is that it contains kind of a combination of compounds, right? Rosemary, ashwagandha, and luteolin, and this activates this antioxidant system, NRF2.
Yeah.
What makes this different from any other antioxidant activator? One that people talk about as activating NRF2 is sulforaphane, like a broccoli-derived compound. There's vitamin C and vitamin E and other antioxidants. What makes this different from all the other antioxidant activators or supplements that are out there?
I may answer this question for you because in past about 12 years, and we're screening a larger number of plant-derived compounds, and both individually and in the combination to see which worked, the best in activating the NRF2 pathway. What emerged was that, in PB125, we have three botanic compounds, and from rosemary, ashwagandha, and luteolin. Each influence NRF2 through the complementary mechanisms. So when they combine, they produce a synergistic effect, meaning that the overall activation is greater than the sum of what we expect from each ingredient on its own. So through this screening process, we formulated the PB125, as you probably see on our label of PB125. And the ratio of those three is 15:5:2. And this formulation consistently produces the strongest and most reproducible NRF2 activation in our laboratory. And that's why we picked that formulation. So the sulforaphane works a little bit differently, and it's primarily a single electrophilic nerve-2 inducer, and that activates the pathway by just modifying the KEAP1, and that protein in the cells that normally keeps the nerve-2 in check. PB125, by comparison, appears to influence the multiple regulatory control points within the nerve-2 pathway rather than rely on single mechanism. So that broader approach may produce a more robust and potentially more sustainable activation of nerve-2 network in our cells.
Super interesting. Clearly, this is a pretty interesting way of activating the body's own antioxidant system, so much so that probably the most well-known longevity interventions testing program called the ITP selected PB125 as a candidate intervention to test. But I know the findings there were a little bit kind of complex to interpret, or I know that you guys had some perspectives on it. So do one of you want to share kind of what came of the ITP studies and what your perspective is on them?
Yeah, the ITP program is, the top kind of mouse models approach to studying longevity, with where they take in candidate molecules to study. And the first paper out of that was basically based on log rank testing, and where there was not a beneficial effect seen. But there, again, this is all third party. We were not involved. We just supply reagent. But then Zhang and colleagues developed a newer statistical analysis approach, which they called the temporal efficacy profile. And so when they included time as a function of effects on longevity, there were beneficial effects observed for several. What they weren't just studying are restudying. They were restudying the entire ITP program that way, and several molecules were revealed to have longevity enhancement, geroprotective effects, including PB125. And then it's a fascinating paper to look at, because they also discovered some time frames of toxicity for some other agents that were studied in the ITP program, or negative effects anyway.
Yeah, I was just wondering, did anything go the other direction, right? Like, where some things originally were found to be positive, and then maybe with this new analysis went the other direction. I'll have to read it, but I'm curious.
My best answer is yes. There's some interesting things in there that were, it doesn't turn the world on its head, but there were definitely time periods at which some of the compounds of interest were not beneficial. So where PB125 came out, both males and females, was, significant beneficial effects against mortality at specific age points during the mouse lifetime. No periods of negative effects, but some periods. So we were really thrilled to see the reinterpretation of the data, the reanalysis of the data, to come out showing PB125 being beneficial, at no stage harmful. And especially in the older female mice, it was pretty easy to see the benefit by they call it the TEP kind of statistical approach. So yeah, I will definitely share that with you to read, because there's a lot in there, and there's supplementary material as well. There's a ton of that goes along with the paper. So we were really pleased to be included in the ITP study. It was a difficult time. We were in the middle of the pandemic with multiple labs involved in that study. So it was not the easiest time period to do the work, but it was all done. I think they have 3 sites around the country that they work it in. And so we were really proud to be included in that study, and then pleased that the analysis published last fall revealed geroprotective benefits of PB125 in the mice.
Super interesting. Do you know offhand, well, I guess 2 questions. One is, what were those periods where it appeared to be most effective? I know you said in older age for female mice. Were there other periods that were notably positive for this compound? Other question would be, like, how did the effect size, and again, I'm sure this is all in the paper and we could, just by looking at it, know this, but I haven't seen it. So just for the benefit of me and the audience, how does the effect size of PB125 in terms of geroprotectiveness compared to other interventions like calorie restriction or rapamycin, or things that kind of have a more established track record as being geroprotective in this setting?
I can't answer it. I don't really know how to parse it. The way that's presented is, I don't know how to do the comparison side by side to other compounds. It's more like, did it, and they have a cutoff, did it have longevity enhancement effects in these specific time periods? And it's not so much scored against other compounds as, is it statistically present or not? And then to me, the real takeaway was that different agents can have different effects on all-cause mortality at different time periods in the lifespan. It may be that we could glean comparisons in terms of effect size, but I didn't manage that yet looking at that paper.
That paper, it's very technical. So yeah, the general audience without scientific background, it's pretty hard to interpret it. But I mean, again, like I follow up Brooks, then it is positive for PB125 compared to other compounds on the list. So the bottom line is that the toxicity of our compounds is clearly known, but dietary supplement may have some level of toxicity.
Right.
I mean,
Let me
again,
ask a question. Go ahead.
No, I mean that again, this is just a simple test and for the animal model and the dose dynamics of the drug, that it's a complexity of the study. So
Got it.
Yeah, that's one hard part of doing so many compounds in one major study is there's it's not dose response, it's multi-year, and in our case, for a lot of cases, it's added to the chow, which is okay. It's not as representative of daily intermittent consumption as kind of a continuous feed approach. But despite those differences for how at least dietary supplements would normally be consumed, we were glad that we had no periods of negative effects and some periods of longevity enhancing effects during the lifespan of the mice.
So let me ask you a different question, which I know you guys may hesitate to answer, but I think this is the question that people always are interested in. And I will disclaim what you choose to say by saying you guys are not doctors, not medical advice, and so on and so forth. But I think what people are interested to know is, based on everything you see about how PB125 works in animals and in humans, how would you recommend most people use it? And what are the right ages? Like, what are the right protocols based on what you know? And again, I'm not holding you to this being a medical advice. I think like people just want to hear this information, including myself, right? Like, what is your honest perspective on this?
I can give you some general answer for this, but hopefully you go by with this.
All disclaimers are there.
This is really the question, clinicians really wanted to answer. And PB125 is designed for individuals who want to support, healthy aging, by enhance the body's own cellular defense and stress response system. Pretty much it stimulates its own system, rather than targeted as single disease. And it works upstream by activating NOV2. That pathway regulates many of the body's natural protective mechanism involved in oxidative stress, inflammation, detoxification, mitochondrial function, and cellular resilience. And important to emphasize that PB125 is not intended to diagnose, not for treat, prevent, cure any disease, and it should not be reviewed as a substitute for established medical therapies, particularly for conditions such as cancer and other chronic disease. One challenge with interventions that support healthy aging is that biological changes often occur long before they become apparent clinically. At the present, if you want to say, hey, is there any biomarkers we can check for PB125's response? At this point, we don't. But in our research laboratory, we can check to regulate gene expressions and other molecular markers. So that at this point, it still remain research tools rather than the test used in clinical practice. But I think the physicians like yourself can look instead to growing body of human clinical trial evidence. Recently, we completed double-blind and randomized placebo-controlled study conducted by independent investigators at University of Utah for PB125. That study just been published, and that demonstrated that PB125 produced a meaningful improvement in vascular endothelial function in healthy older adults. Study also showed that PB125 helped preserve vascular function during prolonged bed rest in older adults and prevent vascular dysfunction associated with limb immobilization in young adults. Those findings are noteworthy because endothelial function is widely recognized as an important indicator of vascular health and healthy aging. Demonstrating improvement in this well-established physiological measure provide increasing evidence that activated nerve tube by PB125 can really translate into measurable functional benefits in humans. Okay. And that's why additional study may be certainly needed to confirm broader clinical outcomes. Those results suggest that PB125 is more than a compound that simply changes laboratory biomarkers. It is now demonstrates significant effects on human vascular physiology in these rigorously conducted clinical trials, providing physicians with evidence-based rationale for considering PB125 as part of the comprehensive healthy aging strategy for individuals. I mean, in other words, for your patients. So.
Yeah, super interesting. I mean, I think as I just reflect on what you just shared, as well as what we see from mice, like it certainly does seem like older adults, older individuals could stand to benefit from this in particular. And, in looking at the paper that you just shared, the RCT in humans, right? Like, this was just really, 2 weeks of supplementation, just like weeks worth of supplementation where we then saw like a clinical effect, which is, pretty impressive, honestly. I think one of the things that comes up for us clinically is we'll do tests, right? Like organic acid testing is a good example where we will measure oxidative damage in different ways. Like we can measure DNA damage through a marker called 8-OHDG. We can look at peroxidation of lipids, and we can determine that somebody has oxidative stress. And the question then is, okay, what's the best way to remediate that? And in addition to encouraging a healthy diet and an intake of antioxidants through food and through exercise and things like that, people will also reach for things like N-acetylcysteine, probably the most common that's used, to kind of help offset excessive antioxidants, right? And so, is it accurate to say that we can use PB125 instead of things like that? Or how would you think about how PB125 could interact with other antioxidants that someone might be using to address oxidative stress from a scientific perspective?
I mean, yeah, I think that in terms of in most parts, there's some consumable antioxidants that get recycled, which is useful, and they're vitamins for a reason. But in terms of supporting cellular defense mechanisms, I think things like PB125 and other NRF2 activators and exercise and food and sleep are the key things I think I would think of suggesting. And in terms of the recently finished clinical trial, it pointed to, so we like to think in terms of really upstream effects. NRF2 as a transcription factor is acting on hundreds or thousands of genes to help control cell defense mechanisms. And the measurement that this group was making, it wasn't us, this was a separate group. We supplied the PB125, but weren't authors or contributors to the study, which was a good way for it to be handled for us. Their measurement of flow-mediated dilation is also kind of an upstream event, or at least something that can be measured that has an impact on lots of different potential health outcomes. And so we were fascinated with that as a readout because one, it was double-blind, placebo-controlled, and beneficial, but also it can anticipate so many other problems downstream of if you see the problem with flow-mediated dilation and endothelial function. And so, we're not suggesting, like Dr. Gao was saying, like we're not suggesting treating or curing diseases, but there's a lot of illnesses that can be represented by endothelial dysfunction or recognized by endothelial dysfunction. So we were really excited to see the benefits both in normal old people and then older people at age, 67 was the median, I think, for that group, and also in young people who are challenged. So young people didn't see a benefit at baseline, but when they had limb immobilization, which could be, there's a lot of disuse challenges that can be seen in real life for sure, but then also mimicked in the lab, either bed rest or limb immobilization, probably other things. So the older subjects benefited at baseline, and then they also benefited when they were immobilized in bed. But in young people, the challenge of having their limb immobilized prevented the degradation of, or the diminishment of vascular function that was observed in those subjects. So mostly we think in terms of an aging, PB125 around aging, but also in terms of other kinds of health challenges that even the younger group might face. But,
so if I don't feel like exercising and I don't want to move, I can take PB125. No, I'm kidding. But super interesting. Let me just kind of shift to another question that I'm sure a lot of people have, which I think relates to the observation that this NRF2 system, mutants of that are associated with certain cancers. And I think some would say, like, hey, if this thing is turning the system on constantly, isn't there a potential concern that we could make something like that worse or cause it to arise? Any thoughts on that from a scientific or what you've observed perspective?
We definitely...
It's really, really important question actually to ask, and they are very different. As you just described, some we call transient activation and versus a constitutive activation of NRF2. And this is really often misunderstood by the people not, with this pathway. So transient NRF2 activation is a normal physiological response to stress. When cells are exposed to challenges such as you just mentioned, exercise, fasting, or certain dietary phytochemicals, NRF2 is temporarily activated, and you switch on genes involved, antioxidant defense and detoxification, cellular repair. When the stress has passed, NRF2 activity returns to its normal baseline. So that's very, very different from constitutive activation, where the pathway is essentially stuck on the on position. This is often a result of mutation genes like in KEAP1 you just mentioned, or another gene. It's NRF2 itself. It's called NFE2L2, which I found in some cancers. In those tumors, and continuous NRF2 activation allows cancer cells to maintain cytoprotective programs that can support tumor growth and contribute to resistance to like chemotherapy and radiation therapy. So for PB125, and other two activators probably fits into the transient and reversible activation in normal cells. They have not been shown to induce that kind of constitutive activation associated with the gene mutation type of activation. NRF2 biology is really complicated than most people thinking. So yes, NRF2 can, and in some circumstances, promote the survival of established cancer cells. So anyone with active cancer or anyone undergoing chemotherapy and radiation should discuss the use of any dietary supplement with the oncologist before taking them. And the key takeaway is that physiological regulated activation and the permanent genetic activation are fundamentally different biological states. And very important distinction when you interpret some oncology literature and the potential role for NRF2 activating supplement.
Along those lines, would you say that people are taking NRF2 activator, PB125 for healthy aging is this something
Mhm.
that they should kind of take in cycles, right? Where people take it for maybe 3 months and they stop, or maybe they take it a few days a week, like 3 days a week, and leave the other two off. I'm curious, just given what you know about this idea of having some activation but not constant activation being beneficial, is there anything that, based on what you've seen, you might recommend in that regard?
I don't think that the cycling of PB125 taking is going to help much, mainly because, for each individual, you don't know your physical status at any time point. So, yeah, I mean, again, this is a food type of activation reagents, and it doesn't do any harm for any of those.
It's a
So,
pretty rapid transient.
yeah,
I mean, by 24 hours, it's back to.
it's gone.
So,
Mm.
it's like, just to jump in there, one interesting study I can point out that came out last year was in horses who have a tremendous oxidative stress burden because of the oxygen consumption in mitochondria in their muscles. They actually have an enhanced to system, like much higher. It's a fascinating paper for a lot of reasons, including the mechanism that kind of a stop code gets blasted through by horses. So it's worth reading that study. But one of the takeaways was they have a huge amount of capability, and that's because of the power usage that they have to get their muscles go through. And there's no other downstream cancer related effects in horses or anything from just this experiment of nature. So that's an interesting one. And so I like to think in terms of like broccoli, the highest quartile of broccoli consumers in humans doesn't have any additional risk of cancers or anything. So these intermittent activators that we would like Nerf2, like PB125 or broccoli or exercise, don't really fit into the constitutively activated category. But even in horses where it is constitutively activated, there's not. I think there's a lot of other mutations that have to be going on for something to be happening for them.
Hmm. Very
Plus,
interesting. Very interesting.
yeah, that is oxidative stress. I mean, it's a dynamic, right? You don't know when that comes, when that goes, but consistently, take these nerve two activators and at least put yourself in check all the time. And again, everybody is different, and you don't know if your oxidative balance is off and how much is off. So it's a safe check to take it, in other words.
Well, super interesting. I want to be respectful of everyone's time, so I appreciate everyone who tuned in, and especially appreciate you guys for taking the time to speak with us. If people are interested in learning more about PB125 or trying it out for their patients, their clinic, or even for themselves, what's the best way for them to do that?
So our website's easy to find, pathwaysbio.com. So pathway is with an S, bio.com. And they can always search PubMed for PB125 or for Dr. Joe McCord and see what we're up to with our publications.
And also, we have a
Awesome.
lot of scientific information on our website, and it's a really good site to educate yourself, and as well as PB125. And it is very well studied, and especially now, it's the clinical approved some physiological effects on human body. So, yeah, encourage everybody to read that.
Awesome. Well, Brooks, thank you so much for taking some time this morning. And thank you, everyone, for listening. And hope everyone has a great week.
All
Thank
right,
you. I love what you're doing
you
with
too.
this.
Thank you.
Thank you.
Thank you.